The adeno-associated virus, as you've heard from many sessions today, has got a relatively small capacity of approximately just under 5 kilobases, and within this region for the DNA we have got plenty of space for our vector. I put up here a diagram showing the arrangement of the regulatory elements within the expression cassette that we're using in our clinical trial. We're using, that you can see the rep1 gene, which is the coding sequence of rep1 in the middle there, which is 1.9 kilobases. Upstream of that we have a CBA promoter with a synthetic splice site, and that's a CMV-enhanced CBA promoter, very similar to the promoter used successfully in Jean Bennett's clinical trial and that from Bill Housworth. We've also included downstream a WPRE, which is another regulatory element that has been shown in a number of clinical trials outside of the eye to be safe and also improves expression of the gene significantly in AAV vectors. Again, our hypothesis is to try and make the expression cassette as effective as possible so that we can limit the total dose of vector required, because the main concern would be an immune reaction by overdosing the vector. This then will get transcribed into messenger RNA, which will undergo splicing and export into the cytoplasm for translation into rep1 protein. The WPRE is a structure that helps in exporting, or is believed to help in exporting the messenger RNA from the nucleus through a CRM1 export pathway. And when we discussed with the regulatory authorities about using the WPRE, they are very keen to ensure that if we were to use this sequence, that we were to make sure that it was deleted in terms of not being able to express any native proteins. And as I said, this WPRE has been approved by the FDA already and has been used in clinical trials in the United States. So our clinical trial team comprises primarily, apart from myself, Miguel Seabra, who's my collaborator and has really worked on, has been a pioneer of choroideremia over the last 20 years, identifying the gene, describing the mechanisms of protein interaction, and really unraveling the mysteries of the disease. Also, we set it up as a multi-center study. Primarily myself and Andrew Webster at Moorfields Eye Hospital probably have most of the patients, but we're very keen to engage other ophthalmologists and other centres, and particularly I'm very aware that patients who are visually impaired find it very hard to travel long distances for trials all the time. So by having 4 different centres for recruitment of the patients and follow-up, we not only have 4 independent ophthalmologists looking at the data, which is very helpful, but we also limit the travelling required for the patients, because undoubtedly they would require follow-up over many years. Our vector was made in the United States at the Nationwide Children's Hospital in Columbus, Ohio, in the new vector facility there under the